Welcome back to the Vitals Podcast. I'm your host, Chauncey, back to digest and bring you the latest in medical news today. It is Monday, May 13th, 2024, and I have some amazing studies to unpack for you. But before we get to that, an announcement for my listeners on Spotify. On every episode, scroll down to the Q&A section below and drop any topics or questions you'd like to see answered in a future episode, and you just might see it in our new segment, The Vitals. Listen to the end for that. Our first story comes from researchers Ashley Maloney and Sebastian Kopf and their joint research team made up of scientists at both Colorado University and Princeton University, where tools often used in geology may be able to detect cancer on the atomic level. To understand how this tool works, we need to know a little bit of chemistry. Geochemist Maloney puts it like this. Hydrogen comes in two main isotopes, or flavors. Firstly, you have normal hydrogen, the hydrogen that bonds with oxygen to make good old H2O. And secondly, there is deuterium, which is heavier and is outnumbered by normal hydrogen on Earth by a ratio of about 6,420 hydrogen for every deuterium atom. While hydrogen has only one proton and one electron, deuterium has one proton, one electron, and one neutron. Geologists have used this difference to analyze anything from the origin and flow of bodies of water to the temperature of the Earth when a sheet of ice in the Arctic was formed. Okay, so what's the big deal? Well, in the research, Maloney and her colleagues grew cultures of yeast and cultures of mouse liver cells. The idea is that cancerous cells use a process called fermentation while normal cells use respiration. The fatty acids that both processes produce, Maloney's team suspects, could have different ratios of deuterium to hydrogen, which could help differentiate the two processes and, by extension, differentiate a cancer cell from a healthy cell. Here's a brief refresher from biology class. Normal cells in our bodies use cellular respiration to make energy, in which it takes in oxygen and sugar, mainly glucose, and spits out carbon dioxide and water, along with some juicy ATP, which your cells use for energy. Glycolysis, the first stage of respiration, takes this glucose and essentially breaks it in half, creating three products. Pyruvate, the halved glucose, and NADH are ingredients used in later stages of respiration, but it also makes two molecules of that sweet ATP. Although oxygen is used in later stages of respiration, glycolysis itself is anaerobic, which means it does not require oxygen, and it still creates ATP. This is important. Cancer cells, on the other hand, do a lot of glycolysis and then a lot of a process called lactic acid fermentation. This process is also anaerobic, so as you can guess, it is common when there isn't much oxygen to go around, like in your muscles during an intense workout. Cancer cells, however, are known to ferment sugar even when there is oxygen. This is called overflow metabolism. Lactic acid fermentation results in a lot of, shocker, lactic acid, and when it builds up in your muscle cells as waste, it creates that burning sensation. Unfortunately, lactic acids also weaken the parts of your immune system responsible for attacking cancerous cells, which makes this waste a lot more valuable to the cancer survival. In the study, the team of researchers found that respiring yeast cells had a higher ratio of deuterium to hydrogen atoms than fermenting yeast cells, even in the presence of oxygen, much like blood-supplied cancer cells. The researchers also tested this with healthy respiring mouse hepatocytes, or liver cells, and fermenting cells within a hepatocellular carcinoma, or a liver tumor. This produced similar results. Now, while this is still groundbreaking, we don't yet have a way to detect ratios of deuterium to hydrogen in actual living patients, but the potential is still hopeful. Kopf points out that, quote, if this isotopic signal is strong enough that you could detect it through something like a blood test, that could give you an important hint that something is off, end quote. Detecting cancer early is invaluable to treating the disease, and thanks to Maloney, Kopf, and their team, we may have a new amazing tool in the near future to do just that. Our second story comes from Dr. Neil Henderson and his team at University of Edinburgh in Scotland, where the discovery of a new repair cell in your liver could mean more regenerative therapies on the way. The liver has a remarkable ability to regenerate itself. Second to only the intestines, the liver is the most regenerative organ in the body. So regenerative, in fact, that depending on many factors, the liver can be up to 70% damaged and within months fully grow back, even if it may not be at full capacity. Despite this, if damage is too extensive and exceeds the abilities to regenerate, like in acute liver failure or ALF, a patient's only option may be to have a liver transplant. However, this may change in the near future. Dr. Henderson and his team at the Center for Inflammation Research wanted to study how the liver regenerates naturally to potentially innovate a new curative therapy as an alternative to liver transplant. Firstly, they took human liver samples from healthy patients, patients with chronic liver disease stemming from a range of causes, and patients with ALF stemming from either APAP toxicity, overdosing on acetaminophen, what you might call Tylenol, or from hepatitis X, not to be confused with hepatitis A, B, C, D, or E. They then observed the growth of hepatocytes, liver cells, in the damaged area. ALF-afflicted liver samples had more active hepatocytes than the others, prompting the researchers to focus on these samples. Upon observation, however, there was a problem. While the liver did heal some of its damage by replicating cells, what's called cell proliferation, it did not fully heal. To take a closer look, they sequenced genes of the liver cells, creating a pan-lineage atlas of liver cells during human regeneration. Basically, they kept track of the activity of many types of liver cells while the regeneration process took place. This is when they found a unique gene protein, ANXA2+, belonging to the new cell. They then found this same gene expressed in a similar cell in mice and studied its functions. The cell, dubbed the leader cell, seemed to come out to rapidly close the wound before cell replication could repair the damage. This suggests that these leader cells prioritized preventing infection from an open wound in the liver before regrowing the liver. This discovery paves the road for new innovative therapies as an alternative to liver transplants, utilizing these novel leader cells to regenerate a liver beyond the superpowers our body already possesses. Thanks to Dr. Henderson and his team, hope is on the horizon. Now it's time for the Vitals, where I go in depth on what you need to know about topics of your choice. If you'd like your question to be answered, drop it down in the Q&A section of Spotify, and you may see it in a future episode. This episode's vital topic comes from NDS, who would like to know more about psychology and how the brain functions. Well, NDS, psychology is an extremely broad subject. Typically, psychology is defined as the scientific study of the human mind and human behavior, generally of an individual. As for how the brain works, in a nutshell, it receives stimuli, whether external or internal, and reacts. The brain is made up of a lot of cells, and 86 billion of them, billion with a B, are neurons. Each neuron has about 10 to 50 glial cells, which help keep neurons healthy and protected. They create a safe environment for your neurons and clean up any mess. Think of them as the brain support crew, as opposed to your neurons being the main actors. Now, by imagining 86 billion neurons interconnected amongst neighboring neurons, it's easier to see why our brain is often compared to a supercomputer. It's also a control center, calling the shots for almost everything in your body. It does this by sending electrical signals through neurons in your brain, your spinal cord, and nerves throughout your body. And the connections between neurons are called synapses. Synapses transmit signals from one neuron to another through chemicals called neurotransmitters that are released from the sending neuron and bind to receptors on the receiving neuron. You've probably heard of some neurotransmitters: dopamine, your reward neurotransmitter; serotonin, your happiness neurotransmitter; epinephrine or adrenaline, your fight or flight neurotransmitters; and endorphins, your euphoria neurotransmitters. These are just a few. Your brain is split into plenty of different regions or lobes. Starting from the bottom, you have your brain stem responsible for your circadian rhythms, your sleep-wake cycles, consciousness, and your vitals, like your heart rate or blood pressure. Your cerebellum, a bundle of gray matter at the back and bottom of your brain, is responsible for coordination, balance, and muscle tone. Your occipital lobe at the back of your brain is primarily for processing what you see. Your temporal lobe at your temples helps make meaning of what you see and hear. And your parietal lobe at the top of your brain is involved in processing language, touch, and pressure stimuli. Your frontal lobe at the front of your brain handles voluntary movement, attention, short-term memory, planning, reasoning, speech, and many other functions. Psychologists are generally most interested in this lobe as it often controls your behavior. There are countless studies out there analyzing the brain's behavior, like the smoky room experiment examining the bystander effect, or the Ash paradigm examining conformity. If you want to learn more, feel free to do more research. That's all for this week's episode, so thank you for tuning in. Don't forget to follow the podcast and share this episode with your friends. If you'd like your questions answered, remember to drop a response in the Q&A section under this episode. Until next time, keep your vitals strong and your spirits high. This is your host, Chauncey, signing off. Take care and stay healthy. I'll see you next week.